the introduction to the presentation tonight. So just sit tight for a little bit here.
Okay, we are live on Facebook, so your call if you want to wait another minute or two, or if you want to get started. But yeah, we're all good.
Okay, great. Thank you, Teresa. Well, let me once again introduce myself. My name is Daryl Boness. I'm Editor-in-Chief of Marine Mammal Science, and you have joined our Editors Select Seminar Series. We've been doing this now for maybe close to a full year on a monthly basis. The presentations for these seminars come from papers that have been published in Marine Mammal Science and recommended by editors or by myself or anyone from the board of editors for the journal. As I think you probably all know, these are being recorded, so you should just be aware of that. The seminar will be available with a link on our website afterwards. I don't know if it takes a day or two, but it will be up. I should remind everybody that if you have questions for the speaker, make sure to put those in the Q&A rather than in the chat. We'll be monitoring the Q&A for questions to pass on to our speaker. So I guess with that, let me start by introducing Dr. Valentina Melica, who received a master's degree at the University of Trieste in Italy, studying jellyfish, not marine mammals. But she subsequently earned a PhD at the University of Alaska in Fairbanks, where she studied reproductive and stress-related hormones in both blue and gray whales. Valentina is currently a research scientist with the Department of Fisheries and Oceans in Canada. She is part of the Marine Mammal Conservation Physiology program in Vancouver, British Columbia, and her work now focuses on biomarkers in killer whales and humpback whales. Valentina's presentation tonight will be based on part of her thesis work. Will be focused on how hormones, the hormones cortisol and corticosteroids, which are involved in regulating stress responses and metabolism, were impacted by various factors for both blue whales and gray whales. Factors that were looked at are age, sex, reproductive status, season, and geographic location. I guess with that, I also mentioned the paper that this is based on was called Naturally Stressed Glucocorticoid Profiles in Blubber of Blue and Gray Whales in Response to Life History Parameters. It was published in the July issue of Marine Mammal Science. And if you are not a member of the Society for Marine Mammalogy or have access directly to published material in Marine Mammal Science, this paper will be available for 2 months to read freely, so you can have access to it whether you get the journal or not. On that, I'll turn it over to Valentina to give us her presentation.
Thank you so much, Daryl. I appreciate you inviting me to this, and I'm very pleased to share with you all my research. Before we start, again, I'm Valentina Melica. I'm going to start with a little bit of an overview of who I am and where I come from. All right. Okay. There we go. So I was born and raised in Italy. I actually grew up not on the ocean. I grew up in the city of Treviso, which is located about 20 minutes east of Venice. While not on the ocean, there's a lot of rivers and water bodies around. As Daryl had mentioned, for my university education, I moved to the city of Trieste, which is located on the very most eastern end of Italy, on the border with Slovenia. And here I obtained a bachelor in biology and then a master's degree in environmental biology from the University of Trieste. During my master, I alternated periods studying in Italy as well as periods studying abroad, specifically in the United States as an international student. And that's where I started being interested in marine mammals and had some experience with some behavioral studies on harbor seals. Then I went back to Italy to finish my degree with a thesis project with the National Institute of Biology in Slovenia that actually didn't focus on marine mammals, was on jellyfish. So for my master, I study sexual reproductive strategies from different population of moon jellyfish polyps. After working for a couple of years in aquarium marine reserves, I had the opportunity to intern at Cascadia Research Collective in Olympia, Washington. And this is where I really was able to dig a little more and sometimes literally into whales biology. This is also where I decided that I wanted to go back to school, pursue a PhD, and specifically researching marine mammal physiology. So I started to apply to different funding sources, different universities. And in 2015, I was finally awarded a Fulbright scholarship that had allowed me to start my PhD degree at University of Alaska, Fairbanks at the College of Fisheries and Ocean Science. Now, I was based in Juneau, which is a little milder than Fairbanks. And I was able to pursue a research in the endocrinology lab under the supervisorship of Dr. Shannon Atkinson, focusing on reproductive and stress-related hormones in blue and gray whales. And this is what I'm going to talk to you about today on my research that I did during my PhD. Currently, I am a research scientist with the Department of Fisheries and Oceans, Canada. I am talking to you from West Vancouver. I am part of the Marine Mammal Physiology Conservation Lab, and I'm involved in different type of projects that still looks at physiological biomarkers. I'm branching a little more, learning more about lipid analysis, stable isotopes, and looking at different tissue and at different species. I'm now mainly working on killer whales and humpback whales. Okay, this is a little bit about me, and now we're ready to dig a little bit into the research. As Daryl mentioned, we recently published a paper from my PhD work titled Naturally Stressed: Glucocorticoid Profiles in Blubber of Blue and Gray Whales in Response to Life History Parameters. First of all, I'd like to acknowledge my co-authors, Shannon Atkinson, who was my advisor during my PhD from University of Alaska Fairbanks, Jonathan Scordino from the Macat Tribe Marine Mammal Program, John Kambakidis from Cascadia Research Collective, Amy Lang from NOAA Southwest Fisheries Science Center, and Diane Gendron from CCMAR, University of La Paz in Baja California, Mexico. They all have provided samples as well as guidance and support throughout this, not only this paper, throughout my whole PhD. Okay, now let's introduce the whales. So for my research, I focus on two species of baleen whales, for which of them we have recovering population in the Eastern North Pacific. I focus on the Eastern North Pacific population of blue whales and of gray whales. I want to acknowledge the illustration from Uko Gorder that I am going to use throughout this presentation to help you guide on when I'm talking to one species, in relation to one species rather to the other. Okay, as I mentioned, both this population have been recovering after being driven close to extinction during commercial whaling. While they're very different species, they do share some characteristic. In the North Pacific Ocean, in the Eastern North Pacific Ocean, they share quite a bit of the migratory corridor. Here in the map, you can see the migratory routes of blue and gray whales. Blue whales are indicated by the red line. Blue whales spend their winter months off Central America with known reproductive grounds in the Gulf of California. And then they migrate during the spring and summer months along the US West Coast where they form large feeding aggregations all the way to the Gulf of Alaska. Gray whales, we know a little more about their migratory areas. Specifically, we do know that their feeding grounds are in the lagoons around the Baja California Peninsula, and that's where they spend most of their winter months. For them migrating north along the US and Canada West Coast, where they're into their feeding grounds, and some of them then migrate all the way to the Arctic, reaching their feeding ground in the Bering and into the Chukchi Sea. Besides habitat, they have some similar feeding ecology. Both of these species do feed on lower trophic levels. And while blue whales are very specialist and they feed almost exclusively on krill, gray whales are more opportunistic, feeding on a variety of benthic invertebrates. Sharing habitat, they also have some shared challenges. Their migratory corridors highly overlap with some of the busiest shipping and commercial lanes, especially off the U.S. West Coast, which put them at risk of vessel strike and definitely exposed to a lot of noise. Also, they're highly susceptible to entanglement in fishing gears. And finally, being animals that feed on lower trophic level, the prey abundance and availability can be highly affected by changes in the oceanographic conditions. Again, this is a map of the North Pacific Ocean, and here highlighted is the sea surface temperature in 2019 during the heat wave, indicating this temperature to be much higher than any average in the previous year. This was also known as the blob. All these challenges could have serious repercussions on the overall population's abundance and dynamic, and for example, can contribute or can be cofactor leading to what are known as unusual mortality event. Unusual mortality event are usually declared when there's a larger than average number of stranded whales recorded for 1 year, and they have been detected for multiple species, not only in the Pacific Ocean, across the globe. Most recently, there was a UME, a unusual mortality event for gray whales, declared in 2019. Before that, gray whales have experienced another mortality event in 1999. In the light of this, there is an urge to understand how these animals are coping with these challenges, with anthropogenic disturbances, environmental disturbances. Often these disturbances are referred as stressors. So often we want to know how these animals are coping with stress. What is the stress response? And while we do have a lot of information about behavioral changes or migratory changes, we still don't really have an understanding of their physiological response to these challenges. So let's take a look at stress. Stress, by definition, is the disruptions of homeostasis, the alteration of the equilibrium in the body, and all the repertoire of physiological and behavioral responses that the body can use to reestablish that equilibrium. Now, stress is not always bad. We do actually need stress. Many of us are familiar with the fight or flight response, which is part of the stress response, which is the adrenaline rush that make us much more aware. And, especially in the animal world, can make the difference between live or perish. However, when a stressor is prolonged on time or it's repeated, then the animal is experiencing what is called chronic stress. And chronic stress over again, a long period of time, can be detrimental. Chronic stress is mainly dealt with by the adrenal gland pathways. So let's take a look to these pathways. Adrenal glands are glands that are situated just above the kidneys, and they are the final recipients of a metabolic pathway that starts all the way to the hypothalamus, where the stressor is first perceived. The adrenal glands, in response to this pathway, they secrete specific hormones that are called glucocorticoids. The most famous one, and usually mainly referred to as stress hormones, is cortisol. There are others, and for this study, I chose to actually look at multiple hormones, and that's included also another hormone that is very similar, and that is corticosterone. But what do these hormones do? Hormones act as messenger in the animal body, and they make sure the job gets done. In particular, the job of cortisol and corticosterone is to keep the body going, to make sure there is enough energy when the body is experiencing a higher than usual energy demand. And to do that, they trigger a process called gluconeogenesis, which is the production of glucose, which is our energy coin, from substrates that are not carbohydrates. So cortisol and corticosterone promote the breakdown of fat, promote the breakdown of muscle proteins so that they could act as substrate in gluconeogenesis to make more energy. Over time, these can be very detrimental to the organism and can highly affect other physiological processes, can lead to can affect the immune response, or in some severe cases can negatively impact reproduction. Reproduction becomes a non-necessary process at the time, so it gets shut off. Hormones, cortisol and corticosterone, can be virtually measured in every tissue. However, these two hormones belong to a specific class that is called steroid. And steroids are lipophilic. That means they do love fat, which makes whale blubber a suitable tissue for this type of analysis. There are multiple papers that have shown the validity and the ability to measure steroid hormones, which are not just cortisol and corticosterone, but also progesterone, testosterone related to reproduction, that are measurable, accurate, that can be measured and detected in whale blubber. From the whale blubber that we have here, this is a whale blubber from a stranded, actually, gray whales. Hormones can be extracted using a stepwise process that involves multiple chemical reagents. And then they can be measured using this technique, which is called enzyme-linked immunoabsorbance assay, or short ELISA. So let's take a brief look to this technique. ELISA is based on the antigen-antibody competition. So we have our plate, the 96 well plate. We have our well. Each well is coated with an antibody. We add our sample, which is our unknown antigen, and then we add a known amount of another antigen that is very similar to that hormones. Those two compete for space. They compete for binding to the antibody at the bottom of the well. And when they bind very solidly. Then using a substrate and a spectrophotometry technique, we can actually measure the concentration of our unknown antigen as in relation to the absorbance. Usually the less color, so the lower the absorbance, the higher the concentration, the more color, higher the absorbance. I might be getting this opposite. I'm so sorry. Higher the absorbance, the lower the concentration, lower the absorbance, higher the concentration. But so this is a very suitable technique that is widely used, and there are commercially available kits that are designed to being able to measure different type of hormones in a variety of wildlife species. Now, before we can actually test for stress, and therefore, especially when we want to know about response to an anthropogenic stressors, it is important to have some baselines. That is, we need to know how these hormones, how these glucocorticoids naturally, what is their natural profiles in a life of a whale. And this was the main objective of my study, was to provide baseline hormone profiles in response to, let's call it, naturally occurring stressors, such as growing up or reproductions, or being in the reproductive grounds compared to feeding grounds. So the specific question that we wanted to answer through this research was, do glucocorticoids change in response to sex, age, reproductive states? And then are they different when the animals are sampled in their feeding or in the breeding grounds, or across over time if they're in their feeding season? So for this analysis, we were able to access archive biopsy samples that have been collected since 2002. Specifically for gray whales, we analyzed 103 biopsy samples that were collected all while the whales were always in their feeding grounds. Nevertheless, we were able to divide the samples in two seasonal bins, one representing the beginning of the feeding season, so during the spring and summer months, and one representing the later feeding season, that is in the fall and winter. Then we use sighting history information from photo ID catalogs to categorize the whales based on the age class and reproductive states. For age class, any whale with a sighting history of at least 8 years was considered an adult, and any whale with a known age that was less than 8 years was considered immature. Similarly, we use sighting history as well as probability of pregnancy from progesterone concentration to determine reproductive states for females. Lactating females were females that were sighted with a calf with a smaller animals during the year of sampling. Pregnant females were females that were sighted with a calf the year after sampling. And then using the probability of pregnancy based on the progesterone concentration, we were able to determine presumed pregnant and presumed non-pregnant females. For the gray whales, I want to acknowledge that the samples were collected from the Macaw Tribe Marine Mammal Program, NOAA Southwest Fisheries Science Center, and Cascadia Research Collective, and provided for this work. For blue whales, we did something similar. In this case, we analyzed about 77 samples, and we were lucky enough to be able to gather samples from the different migratory grounds. Some samples came from the US West Coast, which is where they form a large feeding aggregation, and the majority of samples came from the Gulf of California, which is one of the few known reproductive areas for this species. Similar to gray whales, we use photo ID catalog, sighting history information to divide the animals based on age class. And then for females, we use, again, sighting history information and progesterone concentration to determine their reproductive states. And I want to acknowledge NOAA Southwest Fisheries Science Center and Cascadia Research Collective, as well as CCMAR, that have been collecting the samples, that collected the samples and provided the samples for this analysis. So what do we do with this data? So for the statistical analysis of this data, what we wanted to test, was to understand how specific parameters, life history parameters, may affect hormone concentration. So we use generalized linear models to test hormone concentration in response to age, sex, reproductive status, and season or geographic area, as well as a combination of two or more of these factors. Once from the output of the GLM, we selected the 3 best models, and then we use ANOVA and student t-test to determine statistical significance. Okay, on to the results. So for gray whales, we found that cortisol was significantly affected by reproductive states. This is a graph that shows the different reproductive states females group. And what we found was that lactating females had significantly higher cortisol than any other group. It is interesting that we did see elevated cortisol also in calf, and this is likely due to the offload of these hormones during nursing. For corticosterone in gray whale, we found that this hormones was significantly affected by season and sex, and specifically we observed that males sampled later in the feeding season, so in anywhere between September and November had much lower concentration of these hormones compared to animals sampled earlier during the feeding season. This might be reflective of their ability to feed and thus to restore their energy, leading to a decrease in hormone concentration. So let's take a look now at blue whales. For blue whales, we were able to measure corticosterone only in female. And we observed again that this hormone was significantly affected by age and reproduction. We observed that immature, again, we have here in this graph all the different reproductive group for female blue whales. And we observed that corticosterone levels were much lower in immature females to any other of the adult group. Among the adults, we did observe higher elevated concentration in lactating, pregnant, and presumably pregnant female, similarly to what we see in gray whale. When we tested for geographical location, we did not find any significant difference. In this case, we tested for cortisol, which is the other hormone that we analyzed, and we were able to detect and measure in both females and males. And we did not quite observe any statistical difference. However, we did, our models seem to indicate that females sampled in their feeding grounds, so off the U.S. West Coast, or where they're mainly feeding, did show lower concentration of this hormone. So, what are the two main take-home messages for now? First of all, two hormones might be better than one. There's a lot of literature and a lot of growing literature that evaluate stress response and look at hormones, especially in marine mammals. Often we tend to look at just one, and often that one is cortisol. However, there is a benefit in considering more hormones at the time. Both cortisol and corticosterone seems to be measurable and detectable in both blue whales and gray whales blubber, and they can provide complementary information in response to the different factors. Also, I want to acknowledge this was part of a broader analysis that actually also looked at other hormones in addition to this, and putting together a hormone panels actually might be a better way to get a better understanding on the metabolic and physiological status of the animal. For example, especially when we are considering also reproductive related hormones. Overall, endocrine analysis can become a powerful tool to better understand the long-term effects of change and how those can affect survival and reproductions. So, what would be the next steps to finally to better understand stress response? This was again the second part of my chapter PhD on stress related hormones, but we brought this further, and we wanted to find some stressed whales and compare that to healthy whales. To do that, we were able to actually analyze some stranded samples. So we built a comparison between our biopsy samples, with our biopsy whales that we consider as healthy, and then between stranded or stressed whales. We were able to access 3 samples from 3 blue whales that stranded. All these 3 were had a cause of death of ship strike. And then for gray whale, we had a bigger sample size with a variety of different cause of death involved. But this is what I found. Generally, stressed animals or stranded animal showed much higher concentration of cortisol. I would like to point out, here are the healthy whales. Oh, so sorry. Here are the healthy whales, and those are all the biopsies that I show results before. So that include the pregnant females, that include the lactating females, those whales that had elevated concentration of hormones compared to any other group. And as you can see in these graphs, at least for the gray whales, which is the graph to the left, they're much, much lower compared to the levels that we detected in stranded gray whales. In blue whales, our sample size was much limited, and definitely we observe a much bigger spread, but generally we did observe higher concentration in stranded animal as well. As I mentioned earlier, we had a bigger sample size for gray whales. So for gray whales, and especially for stranded gray whales, we were able to also evaluate difference in hormones across different causes of death. Again, this is a graph that includes our healthy whales. So these are our biopsies. And then we have all those 32 samples from stranded animals. And all this divided by the different cause of death. Entanglement, trauma, this is mainly ship strike. Nutritional stress indicated all the whales that were cited as emaciated. Some were collected during the 2019 UME, the unusual mortality event, and some perished due to predation, for example, from killer whales. And what we observed is that animals that perished due to trauma or ship strike had much significantly higher concentration of cortisol. And this makes sense. Likely, when a whale is ship strike, likely might not die straight away. And so it will trigger a stress response that will lead to becoming very emaciated, very nutritionally stressed, that could potentially result into an accumulation of cortisol in the blubber. And this might be what we're seeing over here. Okay, so why do we care? I know I'm talking to an audience of marine mammal scientists to be, or people that are very interested in marine mammals. So this might be a little redundant, but I want to say marine mammals are a very charismatic megafauna. For many coastal communities, they represent a source of economic profit. If we think about whale watching, that relies on the coastal presence of many species and their site fidelity and returning every year. For many indigenous people along the Pacific rims, whales are an important subsistence food or are culturally recognized, culturally acknowledged. And finally, whales are considered ecosystem sentinels, and they fit very nicely into this One Health concept, which highlight interconnections between human health, animal health, environmental health. Healthy whales means healthy oceans, which means healthy humans. With that, I would like to finish by acknowledging the funding agency that had made my PhD possible and that have supported me through this process, as well as all my collaborators and co-authors. And with that, I would love to hear your question. If you are watching this later, please feel free to contact me through my email in case you have any question. Thank you.
Thank you, Valentina. Sorry, I was having some technical difficulty here, so I got bumped off for a second.
Oh, no.
So let me go ahead. I see there is at least one question in the Q&A. So Teresa, I'll go ahead and turn it over to you for starters and see if we get more questions.
Sure. Yeah, that was wonderful. Thank you, Valentina. Really, really clear, beautifully described. I really understand ELISAs now, and I always appreciate descriptions like that.
Please feel free to let me know. If not, I'm happy to talk about ELISA anytime, but I acknowledge it's not necessarily digestible all the time.
No, that was good. Great balance of those methods, but also obviously the really clear results and very interesting. We have a question from an anonymous question saying, given the high traffic environment and climate change and UMEs and all those external stresses, stressors, excuse me, can you assume that free swimming animals are healthy and unstressed, or do you have to consider that there are existing in a state of chronic stress already?
This is a wonderful question, and thank you whoever asked. I guess, yes, I did talk about baseline, but I guess they are benchmarked, they are baseline to their situations right now. So in some ways, we should account for some levels of adaptations to their environment, and thus to the stressors that they have been exposed now for many decades. So likely, what we can build is a comparison to the levels as they are now, to what they might be in a different situation in the future. Ideally, we would like to go back in time when there was no noise. I think there have been studies, especially during the pandemic, that look at this, and I am very excited to see what the results might have been, as they had the opportunity of test a much quieter environment, whales living in a much quieter ocean for a few years.
Wonderful. Yeah, and arguably also that you are seeing significant differences, even if there is some baseline stress. When you looked at those differences in the other research that you talked about, you were still seeing significantly higher amounts on animals that you could really, really link to stressful events. So yes, even if the baseline is high, these significant stress line events are also adding even more onto that. So
that's right. Yes.
Okay, we have another question that says, have you looked at how long these increased hormone levels last post the initial stress event through subsequent sampling?
I did not have this opportunity, but it would be absolutely something like great to do. And it's very interesting. Blubber tissue, we don't really know what is the snapshot of time that we're looking at. We're likely looking at something that could have happened days to weeks to possibly months before. And also, I would like to, for everybody, I should have said this, I am sorry. When we look at biopsy, we really look at the very surface level of blubber, which likely represents longer time lag. So to go back to the question, yes, it would be great to look at repeated samples to analyze, to look at how long, let's say, we can detect those hormones. However, I might argue that blubber might not be the best tissue to necessarily look at that specific trend. Fecal might be a better tissue for that, as it gives a briefer time snapshot.
Yeah, I look at using fatty acids for diet analyses and blubber. We have the same question that we don't know exactly, we have a general idea of what the turnover rate is and how long they last. We really don't have the studies to be able to say exactly how long those different products last. Okay, we have another question that says, great talk. I was wondering if it would be possible to combine isotopic data and hormone data to see if the stress levels may also be related to shifts in habitat use or diet.
Absolutely. That would be absolutely interesting. I think, this is just a piece of a bigger puzzle. I think combining the informations that we have through isotopic data, through contaminants, through other hormones, really can help to better understand what we're looking at, and what is the actual metabolic status of the animal. So yes, it would be, I think it would be something very interesting to do. And I think there has been a recent paper, because now that I'm diving a little more in the stable isotope world, I have read some research that did combine fatty acids, stable isotopes, and possibly hormones to understand the overall picture.
Yeah, like you said, always better to have multiple sources of data, multiple hormones to look at. You don't want to complicate the picture too much, but it's generally, each of those tells you different things and different pieces of information. So we have another question. I think it's sort of related to what you talked about, but I'll just ask it again. how quickly would you expect to see a stress signal in the blubber after a stressful event?
Yes, I kind of expected this question. As I mentioned earlier, we don't really know how long does it take to the hormones to perfuse from the blood or from the muscle tissue into the blubber. However, there have been some research done, especially on marine mammals under human care. I think there's a paper that looked at stress response in dolphins, and what you could do if when an animal under human care, you can stimulate that process that I showed earlier. Let's see if I can figure the slide. Sorry. So you can simulate this process through what is called an ACTH challenge. And then you can measure the hormones in the blood and then again in the blubber or in the feces to calculate the lag time. And I know this has been done, I believe, in dolphins, and they show that the cortisol levels increase after hours in the blubber tissue. There's also another paper that look at this in bowhead whales from subsistence hunting in the Arctic, and seem to indicate that what we're seeing in the blubber is something that happened days to weeks before that event. Little caveat on that, each hormone is a little different. It's easier to detect are hormones that comes from a physiological process that has been going for a longer period of time. It's easier to detect cortisol if it's reflective of chronic stress. It's easier to detect progesterone that has been perfusing over months in a pregnant whale. It might be a little more challenging if we're looking at different type of hormones that have a shorter lifetime, or they have a more pulsatile profiles. Sorry, I hope I didn't get too jargy on the hormone word.
I think folks who are attending this talk are interested in it in some sense. And you always explain it very clearly, so I think that's great. Okay, we have another question. How quote unquote clean does the sampling need to be? If a sample comes into contact with another sample, for instance, could you still get an accurate hormone measurement?
For blubber, yes. I would say that, you don't necessarily would sample. You would not necessarily, so when you get a biopsy or when you get a piece of blubber from a stranded animals, you would only select a section of it for hormone analysis. So you can do your best if you know that samples have been in contact with some contamination of any extent. You can do your best to limit that part. But I will say, at least in my experience, and at least for blubber, I've never had this problem. I never had a problem of questioning the contamination. I did analyze some samples that looked a little old, or maybe freezer burned, or maybe they had like funky colors, possibly due to storage or something went on in the processing, in the storing time, in the storing process. So that is something that I would be more worried about to be consistent with. I'm sure that for things like fecal samples or blood samples, then you have to be much more careful to not have contamination from other animals.
That makes sense. Yeah, so I guess on that front, you've sort of touched on this, but when you talk about, if you could access historical samples, if someone had some in a freezer somewhere, could you theoretically use them? Are there questions of degradation of these kinds of hormones through time you might have to worry about?
Yes, for sure. I think steroid hormones are very sturdy. So I would not necessarily worry too much about them. I would definitely dig a lot into the literature, be very aware of any problems that have might have been occurred through the years. For example, if the samples were transported from one side of the world to the others, if they were towed for a longer period of time, definitely do some validation process in that. But yeah, there's a lot of samples, there's a lot of archived samples that are available and that could be potentially, there could be suitable for hormone analysis.
Very cool. Okay, we have another question. Can you speak to the potential viability of using breath samples for quantifying glucocorticoid or other hormones? I'm sure you get that one a lot.
Yes. And it's in my head right now because I'm started working on it. I had the opportunity of work on a little on blow, of collecting some blow of blue whales, and of trying to analyze hormones. And on paper, I think it's a wonderful opportunity because it's probably as close as we can get as to have to understand what's the hormone panel of that animal at that very moment. However, the methodology might not quite be there. There's still a little bit of methodological assessments that needs to be done. There's some great resources out there, but one of the main issue that we haven't quite understand how to deal with is contamination from seawater. Not all whales, like blue whales, from what I've seen, from what I've been able to see, tends to breathe, tends to exhale once they're just outside of the water. But for example, humpback whale starts exhaling underwater. So their blow is probably as a good amount of seawater in it. And seawater will dilute whatever hormones you're measuring. So this is a really good question. I'm sorry. I'm gonna get a little greedy on the data. When we do measure hormones out of the spectrophotometers, they're just numbers. They're just number expressed as picograms of hormones per mil. That's what the instrument is measuring. Then you need to correct those data for dilution factor, and then you have to express those data in relation to the amount of samples that you have extracted. That's how you end up expressing hormones as per grams. That is the per grams of blubber that you have extracted, the per grams of feces that you have extracted. We don't have that denominator yet for blow, or at least I might need to catch up on the literature, but I can't recall, to my knowledge, we haven't quite found that denominator. So until we found that, it's a little difficult to interpret those results from the blow. But we're on board.
Stay tuned. It'll come.
Stay tuned. I hope. There's a lot of research going on blow. So.
Very cool. Daryl, did you have any questions that you wanted to ask, since we're on a lull here right now?
Yeah, one question I had, kind of following up on the storage issue and impact of that. For these stranded whales, looking at the picture of the stranded whale you had on your slide, looked like it had been around for a while. How long after being stranded, what kind of carcass condition can you go and still get viable results?
I would say as soon as possible, of course, but I was not picky in my PhD. I do have I had the opportunity, and I still have this data to analyze. I did look into concentration of hormones in response to carcass conditions. However, while the Stranding Network does an amazing job in recording all those data, we have to remember those codes are somewhat subjective. So and if I remember correctly, when I was looking at the data, there wasn't quite a unified code for carcass conditions. So that is tricky to begin with. But yes, it's something that it would be very interesting to test. It would be very interesting to test how like carcass condition might affect concentration of hormones. It might inflate them as a result of metabolic process going on into the blubber as the animal perished, and as the animal as the blubber decompose. And but yes, it's definitely something to keep in mind and something to investigate.
Okay, thank you. Maybe one other question. In a couple of your earlier slides where you had the various categories of animals, I noticed in the presumed pregnant animals, I don't recall if the bars were standard deviations or range on those slides, but it looked like there was a much greater variance in that one group and both those slides. Any reason why that is?
So I'm sorry, you say presumed pregnant or presumed non-pregnant? Or both?
I thought it was in the presumed pregnant, and there were two slides at least where it looked to me like, and I don't remember which slides they were, where that category had much larger bars. Like, yeah, there's one. Yeah.
Yeah, I think it was yes. The blue whales had yes, they definitely had a much larger variability on that. And I'm so sorry, could you please repeat the questions? I've been
well, I wondered if you had yeah, I was just wondering if you had any idea why there was greater variance in that particular category in these couple of slides where it looked like that was the case.
Yes, I think so. One thought is presumed pregnant is based on probability of pregnancy calculated based on progesterone. However, not all these whales were successfully able to complete their pregnancy. Some of these whales, and I might refer you to the paper on this specific topic, did not show up with a calf the following years. And it's possible that they experience, we don't know, of course, there could be a numerous reason why that's so, but one of them is they experience reproductive failure. So if they were experiencing reproductive failure, it's possible that's why they had much higher concentration of corticosterone, or another possibility is that could be depending upon their body condition during their pregnancy. One other thing I want to bring to this discussion: corticosterone is not very within the metabolic pathway of steroids. Corticosterone and progesterone are very close. So there's a chance of some transition between one hormones to the others. And thus, if the whale had much higher progesterone, much lower progesterone, that might have turned that might be metabolized to some level and might be metabolized into corticosterone, affecting the actual level of the hormones. I'm sorry. I don't know if I responded to your question.
Yeah, well, I'm not sure there was a clear response. I was curious to see what your thoughts were on that. So I don't see any further questions. So Teresa, unless you have any, I think we've come to an end on this.
Well, Daryl, I'll ask the question that we like to ask of students that I told Valentina about, for students, I should say, especially for someone who has, successfully completed a PhD and gone through all these different experiences, and as someone who clearly started in a very different field in undergrad, right, and sort of found this interest through time, I was wondering if you have any particular advice that you would want to share, or experiences or things that really stood out for you. They're like, yeah, this really key to getting me to the point that I'm at today?
I would say don't give up, which I realize it might not mean anything, but really, I had so many people, so many times, either telling me that no, not really, it's not going to be a useful field, or you're not going to find a job, and you know, all those kind of things. But really, this is what I want to do. And if I can do the best that I can to do that, I will. So perseverance is the key. I want to share this. I did a PhD as an international student in the United States, and it wasn't always easy. I struggled with funding. I was a teaching assistant for the majority of the semesters of my PhD, which is it's a very skill to build, but it's also very time consuming. So it comes with challenges. But I am glad I didn't give up. So don't give up. And believe it or not, a lot of other people are dealing with the same struggles that you are right now.
I feel like that's a great, oh, sorry, go ahead, Daryl.
I was just going to say, I really like your answer to that, and I'll give you kind of a follow-up. So I've been in this business of research on marine mammals for 50 years, and I can recall when I was working on my PhD on gray seal reproductive behavior, my parents asked me, well, what are you going to do when you finish? And I didn't know. I honestly hadn't even thought about that. I was just doing what I wanted to do and loved it. And so I gave a kind of almost flippant answer. Well, I could work for the Smithsonian Institution. Well, in fact, my entire research career was at the Smithsonian Institution. So you just never know where you might fall into. So I like the idea of just follow what you want to do and persist. And of course, 50 years later, I've done a few other things, but you'll find the right thing. So not everybody does, and you might move on, but persisting at your stage especially is important.
Yes. One other piece of advice, if I may, is don't be afraid to change. When I started my PhD, I thought I was going to work on seals. That was what I thought. I thought I was walking into seals organochlorine research analysis, contaminants. And the more I read, about hormones, that completely switched. And I was very lucky to have the opportunity to talk to my advisor and be like, can we explore different type of projects? And yes, it was stressful to start a PhD and within 6 months, not really have a project, but just a vague idea of what I was going to do. But at the same time, it was very beneficial to me to take that time to do my literature research, to dig into the literature, to understand exactly what I was interested in, what, the availability, where was the niche of need, and actually going for that. So be surprised. Great. I guess I was very surprised. I ended up I had the privilege to see a blue whales, and not just one, a few blue whales, which is amazing, which is one of the best memory of my life, really.
I think that seems like a great point on which to end things with some fantastic advice.
Well, do I see a question popping up in the Q&A?
We have an expression of thanks.
Oh, you're welcome.
It was a absolutely wonderful talk.
Yes, on that, I will thank you, Valentina. I agree. It was a great talk, very clear, well presented, nice job of answering questions. So thank you very much. I appreciate a great presentation.
Thank you. Thank you very much. Okay.
And as I end all of these, keep marine mammal science in mind for future publications.
Absolutely. Absolutely.
Great.
Thank
Okay,
you.
well,
Thank you.
You're welcome. With that, we'll go ahead and sign off. Thanks to all who joined our presentation tonight. And if you missed it tonight, look for it on Society for Marine Mammalogy website to watch it later. All right. Thank you much. Good night.
Thank you. Have a good night.
Have a great evening.